Hi, everyone. Welcome back to the MBP podcast. My name is Calder. I'm here today with Dr. Bojana Stefanovic, the Physical Sciences Platform Director of Sunnybrook and also Senior Scientist at Sunnybrook. Dr. Stefanovic, thank you for being on the podcast today.
It's my pleasure. Thank you for inviting me.
So can you first talk about sort of your background and the path you took to become a neuroscientist in Toronto?
So I did my undergrad training in electrical engineering at the University of British Columbia in Vancouver, BC. And then I moved to Montreal, to McGill, to do a PhD with Bruce Pike at the Montreal Neurological Institute, studying new ways of imaging human brain function non-invasively and trying to extract physiological variables that are changing when the brain is being activated. And then I did my postdoctoral fellowship at the National Institutes of Health in Bethesda, Maryland, under the supervision of Afonso Silva. And this had me move to kind of preclinical models of brain diseases and also in preclinical imaging period using MR, but also other optical methods in electrophysiology. I've done a research fellowship at NIH as well before taking on the faculty position at Sunnybrook Research Institute.
So I guess what's your day-to-day life like as a scientist at Sunnybrook?
So it has changed a lot through the pandemic years, but it's day-to-day, a lot of interactions with bright, enthusiastic young people, which is what brings me to work. And then science talks with collaborators, a lot of grant writing and paper reviewing. And on the whole, it's really having fun studying brain function.
So I guess we mentioned Sunnybrook a couple times here and in previous podcasts as well. So I guess, can you just talk about Sunnybrook Research Institute in general and how it's related to UToronto and medical biophysics?
Sure. So Sunnybrook Research Institute comprises three platforms: physical sciences platform, the biological sciences platform, and evaluative clinical sciences platform. And most of the medical biophysics scientists or appointed faculty who are at Sunnybrook are in physical sciences platform, and our former physics stream faculty, the majority of them from the MBP perspective, also are in the physical sciences platform. At Sunnybrook, we have thus had, I guess, from day one of SRI, which precedes my time, a very strong bond to MBP. Much of the research and much of the teaching, in fact, for the physics stream students over the years has happened. And Sunnybrook, so there is a long list of faculty in the platform and associate scientists as well that are engaged in development of medical imaging and medical technologies that are MBP faculty who are working here under very excellent infrastructure.
I know there's also like a bunch of faculty who are now working at Sunnybrook who got their PhD degree at UToronto through medical biophysics, right? I find that really interesting.
We have homegrown faculty as well.
So I guess, can you talk about your general area of research and some exciting projects you're working on?
Sure. So as I mentioned very briefly previously, my research interests are in imaging of brain function. And we're specifically quite curious about how brain function gets implemented specifically on the level of interaction between neuronal network and vascular network. So the basic computational unit in the brain is the neuron, but on its own, a neuron, I guess, does very little. And when it comes to our cognition, when it comes to our behavior, all of those fabulous functions that the brain is able to perform, they're all implemented through essentially coordinated and distributed patterns of activity in the neuronal network. For that to happen, however, neurons have to signal to other brain cells, most prominently to astrocytes and to vascular associated cells, to support themselves in terms of chemistry, but also in terms of metabolites and clearing of metabolic wastes. And vasculature is, from the imaging perspective, a more accessible target than the neurons themselves are. So in a lot of our studies, in a lot of the imaging modalities that are interested in quantifying brain function, trying to study how it changes over development or aging or in disease, what we're really doing is making inferences about neuronal activity based on measurements of brain vascular state. And so that's something that we've been studying in one form or the other for a long time, in both clinical research, but also in many experimental models of disease. And specifically at Sunnybrook, we've been interested in models of Alzheimer's disease and focal ischemic stroke. More recently, we've added mild traumatic brain injury to our list of model diseases that we've been investigating and trying to make sense of with respect to this coupling between neuronal and vascular networks, and how do they get changed in the injury, and then what might happen to them over the course of recovery, and how is it that we might be able to use imaging to both understand what's going on and potentially identify things that we might be able to tweak to enhance endogenous recovery post-injury.
So I guess on the more vascular side, you're doing some work in 2-photon fluorescence microscopy. So I guess can you talk about some sort of novel developments that are going on in your lab that sort of make your approach on this problem unique?
Right. So 2-photon fluorescence microscopy has been around for decades, and it's a very exciting method when it comes to getting cellular scale images of the brain, of a live mammalian brain in situ. So in animals that are either anesthetized or awake, acclimatized to imaging. It allows us, because of the plethora of different transgenic models, mostly in rodent and small animal models, to look at individual brain cells and changes in their functioning in real time, image them, and then do quantitative analysis of what's going on functionally, and how might these injury models that I mentioned before interfere with their functioning. So in terms of the recent developments, vasculature has been something that people have been imaging for a long while using either mutant models that express fluorescent reporters in endothelial cells along the vascular wall, or they've been injecting large molecular weight dextrans, which in the presence of intact blood-brain barrier stay intravascular. But more recently, a number of very large-scale initiatives in the US have delivered voltage-sensitive fluorescent reporters of neuronal membrane voltage. And these are really, really exciting to us, as they are to many other people, because they offer an imaging glimpse into actually changes in neuronal activity in vivo in real time. So basically, the level of fluorescence emitted by these reporters is a measure and a reflection of the neuronal membrane voltage, in other words, really neuronal activity. So we have various projects right now trying to further develop this technology and also apply it in our experimental models to study changes in neuronal excitability and tone in the context of brain disease.
Definitely heard some work about using like fluorescence as a marker for something else. So it's really cool to see, I guess, as an electrical marker for voltage. That's a super interesting technique there.
Yeah, it gets particularly exciting, I think, when you combine it with all the optogenetic models, where through either manipulation, transgenic models, or through introduction of adeno-associated vectors with specific payloads, you can introduce a light-activatable channel into neuronal membrane. So what then happens is that, such as channel rhodopsin, most famously, but others as well, what then happens is that by using select frequencies of light, you can both perturb the neuronal membrane voltage, and you can then watch it using these fluorescent reporters that I just talked about and see how it's changed. So the 2-photon fluorescence microscope becomes a platform for both imaging, but also for modulating the state of the brain. And that has been something that we've been working very hard on deploying in these models.
Yeah, so my research is actually involved in the heart, but part of my focus as well is correlating electrophysiological data and MRI data. So I was wondering on your side, like, how are you sort of correlating the, I guess, electrical data from, say, EEG with the fMRI data of the brain to sort of get an idea of the crosstalk between the electrical and the functional activity.
Right. So this is something that we've been, I think, early on interested in trying to develop, if you want, bottom-up models of some of the functional MRI signals in terms of how does a certain pattern of neuronal perturbation translate into a certain pattern of changes in the functional MRI contrast. We have, even though we still use MR a lot and still use electrophysiology a lot, our focus has actually shifted towards thinking of the changes in neuronal and vascular network as, particularly in the disease, as coupled, but not as tightly as they are under physiological state. So a lot of the studies nowadays that we run, which employ both electrophysiology and functional MR, we are not so much trying to correlate the two signals as understand how are the changes in the two actually discrepant. So most of the time we're actually interested in those cases where as a result of either injury or response, brain's endogenous response to that, they start diverging, right? And neurons start doing one thing in terms of the electrophysiological signals, and your vascularly weighted MR signals start doing another. So we're really curious about that, and especially in the context of disease, like I said.
So what are some of the recent breakthroughs of fMRI in the sort of neuroimaging setting for stroke and other psychiatric disorders that you're interested in?
To be honest, I think, at least my personal bias has been that one of the more exciting recent developments has been this shift away from trying to use fMRI to identify distinct cohorts towards asking what would it take in terms of the MR protocols for us to be able to identify, so diagnose and potentially predict response to therapy or chart the response to therapy to interventions on a level of an individual. And this is something that's very easy to say. I just said it took no effort, very hard to do, as a result of a lot of heterogeneity, a lot of the background noise, I think, that and variability in terms of how, I guess, brain function gets really implemented in terms of nuts and bolts in individual subjects. And how is it that injury or a progression of disease actually modify the different brain cellular populations? So getting from cohort contrasts to individual assessment, and if you want precision medicine on an individual basis, I think is the big frontier right now for the fMRI, and something that many scientists are working on around the world.
Yeah, I mean, fMRI is definitely important to have good protocols and good analysis on. I think there have been some studies that show that you really have to correct for some of these factors to make sure your results are even usable. Yeah.
Mm-hmm. There are a great many considerations. I like to tell my students that one of the challenges we're daily faced with, not just with fMRI, but with our other imaging modalities when it comes to brain function imaging, is that the signals we're interested in are very small on top of a very strong baseline. And when the effect sizes are not small, so if you get a very large effect size, that typically is fatal to the mammal. So necessarily, if you're studying a disease in which the mammal is surviving over a protracted period of time and their behavior and cognition are slowly changing, then the signals you're looking at are really puny, which makes it a technological challenge to do well.
Absolutely, yeah. So I guess pivoting more to sort of your, lab environment, what kinds of qualities would you look for in a, say, potential grad student in your lab?
So I think above all, what I try to get a sense for in talking to prospective students is the level of their curiosity, innate curiosity about the brain. I think when I started doing my graduate program, and this has been true also with many of the other trainees I interacted with over the years, many of my friends and colleagues, we're really driven by a keenness on finding out how brain works and what goes wrong. And I find that if you come with it, it's something that can stay with you for a very long time and also help you get through a lot of disappointments or maybe failures or maybe things not going quite the way we wanted it to. Brain is a very challenging organ to study, but at the same time, phenomenally fascinating in terms of its diversity, in terms of its resilience, in terms of its redundancy. And it's just really, also from an engineering perspective, something that I think appeals to a lot of engineers, in that it's the control system for the body. So how does it work? How does the nature pull it off? I think it's that innate curiosity that I'm really keen on people having when they apply.
So I guess, could you talk a little bit about your style of mentorship for your students and making sure your students can sort of move forward with their project and, well, eventually after their degree, become successful scientists?
Right. So what I try to focus on early on is, I guess the crass term to put it would be the management of expectations. So I find undergrad research projects and laboratories are very often geared towards executing on certain number of steps. And if you do it carefully, and if you do it in order, and if you cross all the T's and dot all the I's, the outcome is pretty much a guaranteed successful outcome. What I try to tell students from day one is that graduate research is nothing like that. And whenever we are doing something interesting, and hopefully that's most of the time, we're doing things that haven't quite been done before. And we're trying to truly experiment. In other words, try something novel that may or may not be feasible. And even if it is feasible, may or may not produce the outcome we're looking for. And what I try to encourage them to do is develop this perspective where it's when things don't work out that we tend to learn the most. If I hypothesize A and A turns out to be exactly supported by experimental evidence, in some ways it's actually disappointing because I've guessed it and I was right. Where we tend to learn a lot and where we tend to generate the ideas for subsequent studies is that we hypothesize A, but it turns out A is totally wrong and it's actually very different. Not A is true or B, right? Something very different. And that tends to actually generate more exciting research and more exciting projects. And so something to embrace as opposed to be worn down by. In terms of the mentorship style, I find, as I said at the very beginning, very gratifying to interact with keen and smart and driven young people. And so I do meet with everyone in my lab on weekly basis and try to essentially unblock them. I don't look over anyone's shoulder in the lab. I try to steer away from micromanagement, first because I don't have time for it, but secondly because I actually don't think I would be particularly good at it. Mm-hmm. And I think some of my students also think I'm a jinx, so I try to stay out of their way when they're in the lab doing actual experiments. But we do meet on a regular basis to discuss things, and I try to give them a lot of opportunity also to interact with our collaborators, interact with people with complementary and different backgrounds. Because again, it's through these stretches, when you stretch yourself and try to understand a different field's perspective and approaches, that you tend to learn the most about the problem and generate the most creative ideas about what to do next.
Cool. So I guess for students in your lab, or even grad students in general, it's definitely can be hard to find motivation to continue working on our project, and if we hit 5 dead ends in a row. So I guess, what are some things that motivated you during grad school? And also, I guess, some advice you would give grad students to stay motivated, I guess maybe particularly in the age of the pandemic, because we're in it right now, but yeah.
Right. So I found that the key for me throughout my graduate program and even today has been the interaction with other somewhat like-minded individuals who are also engaged in research. Whether they be engineers or physicists or mathematicians or computer scientists or biologists or clinicians, our shared interest, our shared drive towards finding out and towards changing the state of the affairs has been something that has been extremely motivating. And in addition to the personal social capital and the networks of support from the family, through all of these engagements, I find has been transformational. And in the absence of the support from family, from friends, but also from colleagues in our pursuit of these lofty goals, I think has been critical. And it continues to be critical. I actually don't think that it has fundamentally changed between, the training portion and then the PI ship.
So I guess outside of your research, what are some of the other, I guess, maybe more administrative things that you're involved with that you're excited about going on in, say, Sunnybrook or the department or whatnot?
Yeah. So one of the things that I've been very involved in for a couple of years now is in the Equity, Diversity, and Inclusion Committee at the Department of Medical Biophysics. And it started a couple of years ago as a pilot. And then we've realized that we have a lot of work to do, and it has really only grown over the years. The overall mandate has been to try to push the way we do things forward in the sense of making them more inclusive. Our research practices, our departmental processes, and also make the distribution of resources or workloads or engagements more equitable and engage a more diverse crowd of people. And obviously we don't have any answers, but I think over the years we have pushed a number of initiatives that have made things better than what they were before. One of my favorite saying is better. So rather than saying, if we can't make it perfect, we're not going to do anything, just say what would be better than the state we're in right now. And so a lot of the ways that we make the policies transparent or try to introduce additional accommodations or train our student population, train our faculty in a new set of considerations. I find we have been at the front edge of doing, and many funding agencies and many other departments and many other institutions have actually picked up the pace since we started these and have joined these efforts, which I think have to be both made on an individual level and also structurally, so in teams and in institutions. And I think we are making good progress. I think University of Toronto's strategic plan attests to how much progress we've made, and many of the institutional hospital-wide vision and mission statements now incorporate sections explicitly stating whether they're a set of EDI-related goals for the next 5 to 10 years.
Yeah, it's definitely important as a department to be considering this moving forward, and especially in time we're in. Yeah, definitely worth spending time and resources on and making sure that was working for the department. Yeah.
And I think the pandemic and the inequities brought about by or accentuated, I would say, by the pandemic restrictions have made it all that much more important that we are engaged in asking these questions and asking ourselves, what can we do better than the way that we've been doing it thus far?
So I guess in what little spare time you have outside of admin efforts, research, and everything else, what are some hobbies you have that you're interested in? So
one hobby that I think it's quite related to persisting when there are many failures, in face of many failures, as you've described, is I love yoga. And I highly recommend it to all of the trainees. My trainees are really sick and tired of my saying this, but I just find it both physically and mentally rejuvenating and an excellent hobby to have. It's also extremely well suited to the current restrictions. You can do yoga with essentially no equipment anywhere. So it's excellent that way. But there are many other things I really, really, really like reading on a wide range of topics. I love baking. I love food. I think pandemic has also made it possible for us to do more of that, cooking and baking than we've previously been able to, and kind of diversifying our portfolios in other aspects.
Yeah, I guess we have, department yoga now. I think it's weekly. We have a yoga instructor, and now it's virtual, but weekly. And I know in the past you'd have in-person yoga classes sponsored by the department. So it's definitely, it's important for us students to stretch and decompress, literally. But so I guess if you didn't end up going to neuroscience, what sort of other careers do you think you may have seen yourself in?
So I also love gardening, and I think one degree I could have pursued as an alternate, I guess a non-obvious alternate to electrical engineering would have been botany. I think the whole concept of also agricultural engineering and trying to even study the cognition in plants is phenomenally interesting to me, and would have been something that I would have loved pursuing. That among many other things, but
Cool.
botany.
So I guess what is a sort of special skill or talent that people either outside of your lab or outside of your family wouldn't know about?
Yeah, so I want to turn around this question because we've been kept talking about things that worked and things that help, and I want to turn it to things that I'm terrible at. Also because I get a much broader range of things to choose amongst, so I have to exert less effort to identify things that I'm bad at. I am horrible at board games. As a result of the pandemic, I've been playing a lot of board games with kids, trying to keep them occupied during the day. And I'm excellent at losing them. I can lose virtually any. And we've played a wide range, most recently, I guess, Dixit and Labyrinth and photosynthesis. And even though they require different strategy developments, I'm a robust loser at all of them. So there, I'm excellent at losing board games.
Again, Dr. Stefanovic is a Sunnybrook scientist. If people want to get in touch with you, how would they do that?
So we have a lab webpage where you can take a look at some of the recent projects that we've been involved in. And also they can shoot me a quick email at bojana.stefanovic@utoronto.ca.
Awesome. So again, thank you for being on the podcast, and we'll see you later.
So thanks very much. Thank you for having me.